Immunotherapy is a treatment designed to boost the body's immune system to fight cancer. I'm going to break down the two main ideas before we get into the specifics of immunotherapy. First, the immune system, and second, cancer. The immune system works to defend your body against pathogens, which are foreign bodies that include bacteria, fungus, and parasites. It is a network of cells, tissues, and organs that work together to protect the body. Parts of the immune system include B cells, T cells, antibodies, and memory cells. Antibodies are proteins that bind only to a particular antigen. The distinguishing characteristics of cancer were established in 2000 by Hanahan and Weinberg in an article in the journal Cell entitled The Hallmarks of Cancer. I'm going to explain 5 of the 10 hallmarks shown here: self-sufficiency and growth signals, limitless replicative potential, sustained angiogenesis, avoiding immune destruction, and genome instability. Self-sufficiency and growth signals. Normal cells need growth signals in order to proliferate. Without them, they cannot reproduce. On the other hand, cancer cells make their own growth signals and are therefore able to divide uncontrollably. Limitless replicative potential. This means that, unlike normal cells, cancerous cells can replicate infinitely. Sustained angiogenesis. Angiogenesis is the formation of new blood vessels. It is triggered by an angiogenic switch, of which is only turned on during embryonic development and wound healing. However, for cancer cells, the angiogenic switch is always on. Therefore, the cancer will have a steady flow of oxygen and nutrients to keep it alive and functioning. Avoiding immune destruction. Most small tumors are destroyed by the immune system. Therefore, the immune system is effective in killing cancerous cells. This hallmark is the basis for immunotherapy. Genome instability. Cancer is an accumulation of rapidly dividing cells. Because they are dividing so quickly, the replication of DNA is rushed, leading to mistakes in the genome. These mistakes in the DNA are called mutations, and enough of these mutations lead to an unstable genome. Now that you know about the immune system and cancer, we can get into some of the types of immunotherapy, like monoclonal antibodies, immune checkpoint inhibitors, and cancer vaccines. Monoclonal antibodies. In the beginning of this video, I told you that antibodies were specific proteins that stuck to a particular antigen. This is used to fight cancer by programming the antibodies to target specific antigens present on cancer cells. These programmed antibodies are called monoclonal antibodies. When these programmed antibodies are implemented into a patient's body, they circulate until they find the antigen that they are programmed for. When they do find the antigen, they attach to it and send out signals summoning the immune system to destroy the cell containing the antigen. Immune checkpoint inhibitors. The immune system must be able to distinguish between normal cells and foreign cells in order to attack the foreign cells and leave the normal cells alone. In order to make this possible, it uses checkpoints. Checkpoints are proteins on the immune cells that need to be activated or inactivated to start an immune response. An example of one of these checkpoints is PD-1. PD-1 acts as a sort of off switch that keeps the immune system from attacking cells with PD-L1, a protein present on both normal and cancerous cells. Therefore, when PD binds to a cell with PD-L1, it is basically telling the immune system to leave it alone. Cancer treatment vaccines try to get the immune system to attack cancer cells in the body. Most cancer vaccines consist of cancer cells, parts of cancer cells, or antigens. When injected into a patient's body, it triggers an increased immune response in his or her body. I hope that you have gained a better understanding of immunotherapy. It is truly a revolutionary technique that harnesses the innate power of our own bodies to fight cancer.